So my question was to explain light-dependent repair, base excision repair, and nucleotide excision repair. So I drew up some pictures to help explain this. So light-dependent repair is the process where pyrimidine dimers can be directly repaired in a light-dependent process known as photoactive repair. This happens mainly in bacteria, some eukaryotes, but not humans. It depends on the enzyme photolyase, which catalyzes the breakage of the bonds between thymine dimers. The really cool thing about this is that the energy for the process is provided by visible light, so that's where it gets its name. So here are my DNA strands, here are my nucleotide bases. DNA can actually get damaged by UV radiation, so here's the damaged DNA right here. Photolyase, which is the enzyme that catalyzes it, binds to the thymine dimer here, and the cleavage of the cross-links between the thymines happens. And then after that, photolyase is released to present a new repaired DNA. So next we have base excision repair. So the purpose of this repair is to correct a single damaged DNA base. So only one base is corrected or repaired. Deaminated or depurinated bases are detected by glycosylases, which is the enzyme that catalyzes this. They reorganize the base and remove it from the DNA molecule by cleaving the bond between the base and the sugar it's attached to. This is involved in repairing bases altered by alkylation, which is the addition of a methyl or an ethyl group, and deamination, which is the removal of an amine group. ATP is what provides the energy for this process. So we have our DNA strands, our nucleotide bases. Here's the damaged base. DNA glycosylase binds the DNA and removes the damaged base. So here's the removed damaged base. It's called an abasic site. Endonuclease then nicks the backbone, so it creates a space here in the backbone. And then DNA polymerase fills the gap. So DNA polymerase is the enzyme that basically resynthesizes the nucleotide base. So it creates this line right here. And then DNA ligase then seals the nick, so it reattaches the backbone right here. So lastly, we have nucleotide excision repair. The purpose of this is to remove pyrimidine dimers and other bulky lesions in the DNA. So it removes more than one base at a time. It utilizes proteins that recognize major distortions in the DNA double helix and recruits an enzyme called endonuclease that makes two cuts in the DNA backbone, one on either side of the distortion. DNA helicase then binds to the stretch of DNA between the nicks and unwinds it. The gap is then filled by DNA polymerase and sealed by DNA ligase. So here are my DNA strands, again, bases. Here are the two bad bases. So a protein complex comes and it basically unzips the DNA and creates a denaturation bubble. Endonuclease then nicks the backbones, so you see the spaces here in the backbone. Exonuclease then digests the bad bases plus other bases that it was nicked with. So here are the digested bases, or lack of. And DNA polymerase then resynthesizes the bases. So you can see the resynthesized bases, but they're not connected yet. And then DNA ligase seals the backbone. So here's the new DNA with the sealed backbone and the resynthesized bases.